OH Boost project, financed by the EU Horizon Europe program. So my name is Tulbini Nana Karasapugavatte, a research fellow at the Estonian University of Life Sciences, and I will be the moderator for today's event. So we have the speaker, Dr. Ali A. Fouladi-Nashta from the Department of Veterinary Basic Sciences at the Royal Veterinary College. Dr. Fouladi has begun his career as a veterinarian at the Tehran University, where he also lectured in animal reproduction for the 7 years, and he then earned a master's degree in 1995 and a PhD in 1999 from the University of Edinburgh, where he was working with Professor Keith Campbell at the Roslin Institute. Following his PhD, he conducted postdoctoral research at the University of Manchester and also the Nottingham University. So he joined the Royal Veterinary College in 2006, advancing to senior lecturer in 2012, and also now a reader in 2016. So today he will be discussing under the topic size-dependent impact of hyaluronan on embryo development and pregnancy outcomes. So Dr. Ali Fouladi, the floor is yours.
Okay. Good morning, or I can say Terry Humicust. Okay, correct. I don't know. Thank you for invitation, and thank you to Professor Fazeli for arranging this invitation. So today I'll be talking about the size-specific impact of hyaluronan on embryo development and pregnancy outcome. Some of the works that we do for part of my research. So just to say some background about the hyaluronan, which is a linear polymer disaccharide, is a member of the extracellular matrix in mammalian tissues, and it is a non-sulfated glycan, or we call it GAG, and its two main components are glucuronic acid and N-acetyl glucosamine. So they form this disaccharides, which can reach up to 25,000 Dalton. And sorry, let me stop that. So yeah, all right. So it can, yeah. It can be as long as that. So these polymers can join together and form this long thread of these disaccharides. And they are produced by three enzymes called hyaluronidase synthetases, 1, 2, and 3. And they produce different size hyaluronan. The largest size hyaluronan, or if I say HA, is produced by Has2, which is up to more than 2 million Dalton, whereas the Has3 produces the smaller size, which is less than 1 million Dalton. And the receptor for hyaluronan, there are two major ones, is CD44 and RAM, which are membrane bound, but RAM is also present in the cytoplasm. And there are other receptors that I have listed here, but there is a constant turnover of the hyaluronan in mammalian tissues through the catabolism by hyaluronidase enzymes, so-called hyals. And there are a range of them are available. Most importantly, in the mammalian tissues is the hyaluronidase 2, which breaks the hyaluronan in different fragments, and I will explain that in the next slide. Yeah, so once hyaluronan is produced by Has in the cell membrane, goes to the extracellular matrix, but then we have the process of recycling that to break to the disaccharide glucuronic acid and glucosamine through this enzymatic digestion. And The first step is through the hyaluronidase 2, which is breaking that to 20,000 Dalton, equal to 50 disaccharides, and then goes to the lysosomes and breaks down further. So what's important is that during this breakage, all of this could have some physiological functions in the body. So, in fact, very high molecular weight of the hyaluronan, which is initially produced by that enzymes, it is has a role more in the physical property of the tissues, and is important for tissue hydration. As we know, all people use that hyaluronan for creams that they use for anti-aging and so on. So these are, in fact, hydrating the tissues. And there are in other tissues as well that are functioning differently, like in the eyes or in the synovial fluid. And mainly it functions like a space filling and hydrating, and it is immunosuppressive and anti-angiogenic. So it is these functions are associated with a high molecular weight hyaluronan. Whereas there are, when they are broken down, small fragments or low molecular weights also are involved in different functions. Somehow is opposite to what we attribute to high molecular weight. So they are involved in cell proliferation. They are anti-apoptotic. They have a role in the angiogenesis and cell migration. And these functions are receptor mediated, mainly through these two receptors, CD44 and RAM. So overall, hyaluronan has the role of that has been identified in many processes in health and disease. But I will focus on oocyte maturation, embryo development, and embryo implantation in my talk. So in our work, initially, we have shown that the hyaluronan system, including has enzymes, are present in the ovarian follicles. This is example of the ovaries. This is a large antral follicle. See the granulosa cells and the thecal layers. You see that it is highly expressed, and the Has2 is expressed there. Has1 is basically, it is not there. It is very low expression, but Has3 has a lower expression than Has2. So they are there in the ovaries, also in the corpus luteum. We see that the Has3 has more expression than Has2 or Has1. So they are in different parts of the reproductive system, that in the oviduct, uterus as well, that in turn I will talk about them. So what is known mainly in the reproductive system, that people who work on the oocyte and embryo know that the hyaluronan is involved in the expansion of cumulus cells. When the LH surges and the oocyte need to be ovulated during reproductive cycle, this expansion of the cumulus cell, as you see here in this cells, that they are expanding, is because of this high molecular weight hyaluronan produced by this cumulus cell surrounding the oocytes. Okay, so what we did in the first basically to examine the role of the hyaluronan in oocyte maturation, we added the hyaluronidase or hyaluronan to oocyte maturation medium, just as a supplement. And we found when we add hyaluronidase, so there is no cumulus expansion, because high molecular weight is associated with expansion. So when Hyal2 breaks the hyaluronan to small fragments, so there is no cumulus expansion. However, this does not affect oocyte maturation. Okay, so oocyte maturation to metaphase 2 stage has not been affected by Hyal treatment. But when we add very high concentration of hyaluronan, they start to reduce the maturation rate of the oocytes. But if we inhibit hyaluronan synthesis by the oocytes or cumulus cells, by using this chemical called 4-methylambeloferron, which is inhibitors of the hyaluronan synthetases, we see with the dose-dependent decline in cumulus expansion. With the higher dose, we have basically significantly reduced expansion of the cumulus cells. And it is when we add the hyaluronan, okay, when we add hyaluronan, we don't see basically much changes when the oocytes are treated with 4-MU. But this 4-MU seems to affect the oocyte nuclear maturation to metaphase 2 at the higher doses, becoming significant. The majority of the oocyte become arrested at the metaphase 1 stage, and they fail to go to the metaphase 2 stage. So, and then interestingly, when we add 4-MU plus hyaluronan, we have basically tried to reverse the effect on the nuclear maturation. So when we add the hyaluronan, the effect could be reversible. And that's quite interesting to us, that I will get back to this towards the end of my presentation today, whether the hyaluronan could be used as a treatment to reverse some of the negative effect on oocytes. So, and interestingly again about that, and when we treat the oocyte during the oocyte maturation with 4-MU, you see that there is no oocyte could be cleaved. Cleavage is significantly reduced, but then no blastocysts could be produced from these oocytes. Okay, and whereas in the control or the HA treatments, we have cleavage and the blastocyst formation. And when we add even Hyal to the maturation, despite no cumulus expansion, you see that it has not affected cleavage. 75% were cleaved, and they developed to the blastocysts as well. So maturation and development was not affected by Hyal. But when we inhibited Hyal, when we see that, then there is no blastocyst is formed. Again, with addition of hyaluronan, we try to reverse the effect of that. So that's quite important. So this shows that the hyaluronan is important for embryo development. In the next series of experiments, we treated the embryos with Hyal or 4-MU, to see whether it is important for embryo development or not. Okay, and surprisingly, we found that when we add Hyal-2, we see significant increase in blastocyst formation, whereas the 4-MU with the dose-dependent effect basically has a negative effect, and in the higher dose, no blastocyst is formed in the embryos. Even when we add the hyaluronan with 4-MU, no blastocyst is formed, or is very low. So, the lesson from this was Hyal-2 seems to be beneficial for the embryo, but inhibiting total inhibition of the hyaluronan is killing the embryos, and the oocyte development is impacted by that. So that made us to follow the impact of hyaluronidase and how it does it and how it works, basically. So we did some staining for different stages of the embryos for hyaluronan. So this green staining shows the presence of hyaluronan in the 2-cell, 4-cell, and so on. The concentration increases as the embryos progresses. And we look at the structure of the hyaluronan system. The genes has 1, 2, 3, and HYAL2. What we have noticed is that the HYAL2 is start to be expressing from the morula stage. So it's not there at the earlier stage at all. This is the data from bovine. Whereas HAS2 is there all the time, HAS3 is there as well. HAS1 is higher and the low in the earlier stage, but then significantly reduces. So this basically increase or starting to be expressed for Hyal in the embryos makes us become more interested to investigate the role of Hyal. And we know that this early stage of the embryos happens in the oviduct when they are the eggs are fertilized and develop to the morula stage before going to the uterus for early implantation. So we also looked at the changes in the receptors for hyaluronan in the embryos. So they are there. RAM is highly expressed in the embryo, and so is the CD44. And interestingly, we found that the Hyal2 it is expressed also in the oviduct and more in the isthmus than the ampulla. So isthmus is the area where the embryo, after fertilization in the ampulla or ampulla-isthmus junction, they progress. So presence of the Hyal or production of Hyal2 by the oviduct seems to be supporting the early stage embryo, as the evidence from in vitro also showed that. So in order to follow that further, so we did more experiments. We have tested different concentration of the Hyal2, 50, 100, and 300 IU concentration to day 2 embryos. They were produced by through in vitro fertilization, and then they were cultured until day 7 to form the blastocyst. What we have found that when we have the Hyal with the 300 IU, the blastocyst rate almost doubled in the presence of Hyal2. And not only that, the embryo quality in terms of the total cell number were also significantly increased, as well as the number of cells in the inner cell mass. So this was the embryos were stained with differential staining to count the total number of cell and cell in the trophoectoderm and the inner cell mass of the embryos. And so again, when we add the 4MU, no blastocysts were formed. And when we add the Hyal2 here, we had almost another 60% blastocysts in another study. When we add the Hyaluvet, which is a hyaluronan, no changes in the blastocyst formation were observed. And hyaluronan plus Hyal, a slight increase, but was not significant. Okay, so this was also repeated in sheep oocytes. So sheep, we have added Hyal to the embryo culture, similar study that we did in bovine, and we see again increase in the blastocyst rate, and the number of the hatching embryos also were significantly higher with the higher dose of the Hyal2. And then we did some in vivo experiment where we infused PBS or Hyal this Hyal-2 or hyaluronan, or Hyal and hyaluronan together to the oviduct of sheep. They were synchronized for estrus and then naturally mated. The mating were confirmed. Then we, on day 2, after mating, we infused into the oviduct. The oviducts were ligated, and to allow that, so the components of what we infused into the oviduct remained there. And on the basically 5 days later, this sheep were sacrificed and then recovered the oviduct and found the how many embryos were there and so on. So we found that in the presence of the Hyal only, or Hyal plus hyaluronan, we have the increase in the number of the blastocysts found in the oviduct. So this confirmed the in vitro results, and also Hyal increased the number of hatched embryos that we found. So it seems that also presence of Hyal not only increases the blastocyst rate, also accelerates blastocyst formation and blastocyst quality, and they can hatch from zona pellucida. And then to investigate how Hyal functions, we added the Hyal again, plus anti-CD44, as you know, as I mentioned, is one of the receptors. And in the presence of anti-CD44, significantly reduced blast rate, and addition of Hyal-2, a slight increase, but was not significant here. But when we did it further, look at that further, and to find with the action of the Hyal-2, so here we had this protein synthase synthesis inhibitor, U0126, basically significantly dropped the blastocyst formation. And when we added that with Hyal-2, again, significantly reduced the blastocyst formation. So we did the Western blot on MAP kinases, and we found that when we add basically Hyal-2, we have significant increases in both MAP kinase 3 and MAP kinase 1, and as here shows in this graph. So that shows that they function through this MAP kinases, and CD44 is important. Then we looked at the RAM, it's another receptor, and interestingly, addition of RAM totally blocked blastocyst formation. No blastocysts could be produced in the presence of antibody against RAM. So it seems RAM is quite crucial for early stage embryo development. So whereas the Hyal-2 had this effect, Hyal-1 didn't have this effect, okay? And even inhibiting Hyal-1 did not have so much differences. So what we have come with the proposal is that, so when we have hyaluronan production by the hyaluronan synthetases in high molecular weight, they are broken down by Hyal-2 to 20 kilodalton hyaluronan, which is, we call it low molecular weight, or very low molecular weight hyaluronan fragments. They can bind to the receptors, CD44 and RAM, and then functions through the MAP kinases and enhance the cell survival and cell proliferation. Okay, and so moving forward, we said, okay, when does the hyaluronidase does it? So what about the fragments of HA, which is produced by the hyaluronan? How do they function? Whether they have the same effect or not. And interestingly, we found that when we add the hyaluronan fragments, so corresponding to the size of HA produced by Hyal-2, which is about 20 kilodalton, we see that they are has significantly increased, and then in the lower dose, as low as 2 microgram per ml, has significantly increased the blastocyst rate and also the hatching rate of the embryos. And these are some of the images from this 2 microgram. Were almost everything become a blastocyst. That was so interesting to watch them under microscope. So then we said, okay, this is quite interesting. So look at the other hyalos that are present in the reproductive system, including the hyal produced by sperm. So sperm, also we know that the sperm head has the acrosomal membrane, and in the sperm membrane we have this SPAM1, which is called also PH20, is the sperm hyaluronidase that helps sperm during the fertilization to pass through this expanded cumulus cells of by the basic breaks hyaluronan and helps the sperm to pass through and reach to the egg and fertilize that. So, and we know that during fertilization, thousands of eggs, thousands of sperm enter into the oviduct from millions that they are released during the mating. And so then whether they have a role or not in the process of supporting early embryos apart from the role in fertilization. So this data that we have shown here shows that adding pH20 as low as like 10 nanogram per ml significantly increase the blastocyst rate. So it is almost more than doubled the blastocyst rate as compared to higher doses. So very low concentration seems to be effective, but in the higher doses, there is not so much differences. And the hatching rate, or the hatched number of hatched embryos, so it's indicating the embryo quality could be improved. So here, then we did a larger experiment comparing all of them together. So HYL2, PH20, and HA fragments, did it all together, and this also confirmed the previous results and shows that the presence of HYL2 or sperm hyal or HA fragments all supports early development of the embryos. And not only that, we also cryopreserved or through vitrification some of the embryos, and we found that such embryos they have very high survival rate after cryopreservation. So they were frozen and then basically a few days later we defrosted and put them back in culture. So it shows that these embryos become not only developing better, also they are more resilient and more basically they can survive better in terms of embryo quality is much better. So we had 52% in the control versus 80 or slightly more in the hyal treatment or HA fragments. So then the next, we did some embryo transfer. So embryos that produced by this very low molecular weight hyaluronan through the or with as a control, no addition, were transferred to sheep that were the recipients were synchronized to day 7 for embryo transfer. The blastocysts were transferred, and here we found that out of 11 ewes that we transferred, 6 become pregnant, whereas this very low molecular weight hyaluronan group, 9 out of 12 will become pregnant. And so these were, remember, these are from in vitro matured and in vitro fertilized embryos. And so during the surgery, that's through the laparoscopic embryo transfer, and we identified also presence of the corpus luteum, and then when we transfer. So the lambs that were produced, they were all in the normal weight range, and they behaved also normally. And these are some of them that we produced. And Tina was my postdoc who did help me with that. So the proposed model for that is the oviduct producing high molecular weight hyaluronan, but the oviduct also contains the Hyal2 that in the isthmus we saw that it's more than the ampulla, but the sperm brings the pH 20. They can break the Hyal to a small or low molecular weight hyaluronan fragments, which can support early embryos through the proliferation and the differentiation to become a blastocyst embryos. And there are many factors, of course, are produced by the oviduct. And I know Adriza has done a lot of work on the oviducts in the past, or I don't know, still doing that or not, but they are a very amazing organ, and they do a lot of things happening there. And a range of these factors, they may collaborate or help with each other, because we know that these growth factors or loss of factors are produced by the oviduct. They could be also anchored to hyaluronan, could be transferred to the embryos through these fractions or fragmented or low molecular weight hyaluronan as well. So such embryos, they have a higher developmental competence, can result in higher pregnancy. So the next question we try to answer is also was that whether hyaluronan is important for embryo implantation, because people are using hyaluronan, especially for human, and they sell that as called that embryo glue, that they use that as basically whether they want to add embryo glue to the embryos or not, and they make a commercial impact, and to see whether really it is functioning or not, or is related to the embryo quality, whether they see the effect on pregnancy. So here, just give you a small background. You're in the vet school, you should know about the embryo implantation in sheep, but just for those that are not veterinarian, say that, so in sheep, we have the embryos. In contrast to human, they become elongated after forming the blastocyst, okay? So become like a tube shape, then become filamentous to allow embryo attachment to the uterus, which is non-invasive, just superficial attachment to structures called the caruncles in the surface of the uterine endometrium, okay? And during the implantation, which is, as I said, the superficial, we have the pre-attachment, and the embryo opposes the uterine endometrium. This is the endometrium of the uterus. There are changes happens at this interface between the embryo and the uterus luminal epithelium, so epithelium of the endometrium. So these are a number of adhesion molecules are forming between the two to allow that to implantation to happen. So some of them are listed here, like osteopontin, and that I will explain that to you in a few slides. So what we have done is we had the sheep, they were synchronized, naturally mated, and then they were infused on day 14 of after fertilization, after mating. They were infused into the uterus by Hyal or hyaluronan or 4-MU. And we had 8 animals per group. 4 were killed on day 17, 4 were day 35. So just to say that in sheep, embryo implantation or attachment happens on day 16. Okay, so here when we treated that, So this is the process that we did during the surgery. So infusion into the uterus. And here shows on day 17, this is the caruncles. And this white slimy things that you see, these are the embryos, okay? That with higher magnification, we can see that there. And so here with the histology, say that the embryos that were attached. So this is the elongated embryo attached to the uterine endometrium. And here this is very nicely attached embryos. And what we have found is that quite interesting, is that when we add inhibitor of hyaluronan, all the embryos were attached, okay? Whereas when we added hyaluronan, no embryos was attached. This is on day 17, okay? And with the Hyal or PBS, or this is a control, 2 out of 3 were attached. So this shows that the same was on day 35, which is basically all of them were attached. And then we looked at the presence of the proper pregnancy marker. We see these binucleate cells that like that are forming in the pregnant sheep. And then one of the changes that happens in the implantation is basically removal of this mucin 1 or MOC1, is that during the pre-implantation, so MOC1 is there, but then it has to remove as a marker of uterine receptivity, and then allows that the integrin and the osteopontin, they link together and bind to embryo to uterine endometrium. That is what is happening during the embryo implantation process. So we looked at the changes in the integrin and osteopontin in these animals. And so here shows that the treatment in the control group with the PBS treatment. So hyaluronan is not there in the luminal epithelial cells, but it is there in the stroma. HA treated group, hyaluronan is there everywhere. With the Hyal-2 and 4-MU, we can see much reduction in that. So here shows again presence of mucin 1 or MOC1 is still present in the control, but in the 4-MU treated one, they are completely removed. Osteopontin, on the other hand, was highly expressed in the 4-MU treated one. So this is this yellow brown line that you can see here nicely between embryo and the uterus, luminal epithelium, and the trophoblast of the embryo. It can see here also very nicely, it is there. Whereas in the hyaluronan treated, the embryo is hanging and is not attached to the endometrium. Okay, so this also is in line with the previous report that people showed that in mice, changes in the extracellular matrix components during this deserialization in the endometrium. And you say that hyaluronan is pre-implantation is very high, but post-implantation completely absent. So in fact, again, another study showed that if the hyaluronan is very high, basically it results in abortion and no embryo could be implanted. So conclusion from that study is that the inhibition of hyaluronan by 4-MU infusion enhances the embryo attachment. And clearance of HA is necessary for embryo implantation. So just the last few slides, in last 4 or 5 minutes, I will show a little bit more about some other finding we found with the HA. So we know that there are other reports from others that HA content of the ovary changes with aging. Here we have looked at the mice ovary with in the young, is it 3 months old mice, and older ones is 2 years old. You see here, look at the collagen content. In the young, it is very low, whereas in the old, it's significantly increased, and it has been quantified in the granulosa, thecal cells, and the tertiary follicles, and on ovarian stroma. You see that in the old mice, all significantly higher level of the collagen, whereas the level of hyaluronan significantly is reduced in the old one. Okay? So considering the importance of hyaluronan, as I have described in the early embryo development, or oocyte maturation and development, it may have an impact on the outcome of the fertility. So here the embryos the ovaries were stained with hyaluronan binding protein, and shows in the vascular tissue of the ovary significant reduction. Also in the ovarian follicles, different stages, we see significant reduction in the intensity of staining for hyaluronan. So just to summarize, we know that the hyaluronan is binding to its receptor, and this binding allows the internalization of the hyaluronan to the cytoplasm, and even it goes inside, could go to the basically nucleus as well as being degraded through the endosomes. But during the process of that small size of molecular weight of HA is produced, they can involve in the range of functions through activation or binding to its intracellular receptors like RAM, which is associated with ERK1 and can control cell cycle and mitosis event. And they are also associated with the cytoskeleton of the cells through this CD44. So since the hyaluronan is quite important, and there are some other studies show that the hyaluronan during the cell stimulation can penetrate into the cells, into the cytoplasm, and has a role in separation. So it goes towards the nucleus and then goes between the chromosomes and helps to separation of the chromosome during the cell division. So it seems that has a role in the mitosis. So what we know about the aging, we know that the aging is associated with the basically loss of reduction in fertility, and with the changes in the oocyte mitochondria, shortening the telomeres, or coenzyme dysfunction in the chromosomes, and also the stable instability of the spindles. And this particular function or changes could be associated with the reduced hyaluronan. So the question is that, does the reduced HA in the age oocyte affecting oocyte and embryos? Is something that we are interested to investigate that further. So with this, I would like to thank the people who have helped me to produce the data I have presented to you. So funding body, BBSRC, also some of that was with the Origio, and now is the Cooper Surgical, and members of my team as well as my collaborator. And thank you. I'm happy to answer the questions. So I stop sharing.
Yes, thank you very much, Dr. Ali Fouladi. Now the floor is open for the questions.
Good. I think I finished on time. That's great.
Yes, yes. Ali Reza, please go ahead.
Yeah. Ali, thank you very much. It was
Hi,
nice
Ali
to
Reza.
see.
Thank
Hi,
you.
hi,
Nice to
hi.
see you.
Nice to see you too. And yeah, no, you speak more Estonian than I speak Estonian. It's congratulations.
So
Yeah.
is it correct to say Aita?
Yes, yes, yes. It's 100% correct.
Excellent.
Thank you. How is the situation with the work that you're doing? Have you been able also to bring it into the market, and is it possible to somehow commercialize it? How it has been the industry relation with you?
No, we had originally done some works with the Origio, but because we, published some of the results, we couldn't basically we go further to that. But they have the products that now contains hyaluronan, and anyway, so we don't have any more connection with them. But I know that they have. They are utilizing this outcome of this collaboration in their products anyway. But we are not involved in that at this stage.
Then it is getting used in the field, but
Yeah.
okay, no, that is, I think that is already quite a very big achievement. There are not so many products in the veterinary and especially reproductive medicine that reach at the end to the getting used really by the patients or even in the biotech. So,
So this is that, as well as the other thing that anyway I don't have time to present, is that we found hyaluronidase also is produced by human embryos during culture. So, and could be used a potential biomarker as well. So we basically showed that there is a good, we collected the culture media and then measured hyaluronidase with concentration by ELISA. But then we looked at this with the clinical outcome after embryo transfer. So those were done by ICSI or IVF, and looked at that. There was significant correlation between, or those were resulting in pregnancy, and positive pregnancy and live birth has significantly higher level of hyaluronidase 2 in their spent culture medium. Okay, and that was quite interesting. But it needed, there are some noises, but overall, there is no perfect, I think, biomarker. There may be a combination of different factors, but this was high. There was a high trend towards that. And this is also something I am interested to explore that further, whether it's going through the microvesicles or not, something that we can maybe work together on that.
Sure.
Yeah.
Thank you very much.
Thank you.
Questions? Just wondering, how's the infertility or the subfertility incident rate in sheep in the region?
Yeah. So anyway, so sheep also have fertility as well as the cow has fertility issues, but they are associated with different reasons as compared to human. And also one of them is because we use this for production, and especially cows, they are producing a lot of milk, and they have been selected for high milk production. And they go to a stage of, we call it negative energy balance after giving birth. It's because the diet that they receive or food they receive doesn't compensate for the whole amount of energy that they require for different purposes, for maintenance, for the production, for the recovery of the uterus or reconstructing the uterus, and becoming pregnant again. Because for farming, it's important that the cows give birth one calf per year. So the expectation in animals for the industry is different from the human. But what is known that they are also suffering from the infertility and low fertility or soft fertility. Some of that is because of the egg quality, and some of that is related to uterus environment that is hostile to the developing embryo. So finding a uterine receptivity marker or other things all would be interesting to animals as well. Sheep also is the same, but because they are in a big flock of sheep, it has not been fully investigated what is the scale of infertility, but there is a level of the early embryo death and also soft fertility in these animals as well. The same is in the other animals like horses. With age, you have this issue that they reduce the fertility, the same issue because we keep horses longer than sheep. And yeah, so the animals also overall suffer from fertility issues, and that has more importance for economical purpose for the farming industry.
Yeah. Thank you very much. Is there any more questions to Dr. Ali? If not, thank you very much, Dr. Ali Fouladi, for your insightful lecture on